Dear students, we are going to discuss about cell cycle regulation, cell cycle checkpoints, their functions and importance. So, cell cycle. What is a cell cycle? The cell cycle or cell division cycle is a series of events by which a cell divides into two daughter cells. These events include duplication of DNA and organelles and partitioning into two daughter cells. Coming to prokaryotic cell cycle. For prokaryotes, the mechanism of reproduction is relatively simple. Their cell cycle is divided into B, C and D periods. The B period starts from the end of cell division to the beginning of DNA replication. This is the growth phase in which the mass of the cell will be increased. DNA replication occurs during the C period and the D period starts from the end of DNA replication and it is up to the splitting of bacterial cell into two daughter cells. Now we will discuss about eukaryotic cell cycle. In eukaryotic cells, the cell cycle is divided into two main stages, interphase and the mitotic phase. During interphase, the cell grows and accumulates nutrients needed for mitosis. Also, the cell replicates its DNA and cellular organelles. During the mitotic phase, the replicated chromosomes, organelles and cytoplasm separate into two new daughter cells. To ensure the proper replication of cellular components, there are control mechanisms known as cell cycle checkpoints in the cell cycle. These cell cycle checkpoints prevent cell cycle progression at specific points, allowing verification of necessary phase processes and repair of DNA damage if any. The cell cannot proceed to the next phase until the checkpoint requirements are met. The cell cycle checkpoints typically consist of a network of regulatory proteins that monitor and dictate the progression of the cell through the cell cycle. Now, let us look into the series of stages in a cell cycle. Actively dividing eukaryotic cells pass through a series of stages in the cell cycle, and they are the G1 phase. During this phase, metabolic changes prepare the cell for cell division. Next is the S phase. DNA synthesis occurs and the genetic material will be replicated during S phase. Next is the G2 phase where the metabolic changes assemble the cytoplasmic materials necessary for mitosis and cytokinesis. And the next one is M phase or mitosis. There will be a nuclear division followed by a cell division during this phase. The period between mitotic divisions is known as interphase. That is, G1 phase, S phase and G2 phase together comprises the interphase. The next one is M phase or mitosis. During this phase, the actual division or duplication of the cell into two daughter cells occur. So, coming to G1 phase, immediately following mitosis, the newly created cells are in the G1 phase. This is largely a growth phase during which there is a lot of biosynthesis of proteins, lipids and carbohydrates. But there is no synthesis of new DNA during this phase. G1 is the longest of the cell cycle phases in many cell types and most of the physiological activity of a cell happens during G1 phase. The next phase is S phase. Following G1, the next phase of the cell cycle is the S phase, during which synthesis of new DNA occurs. The genome is being replicated during this phase. And at the end of S phase, the cell will have twice the normal amount of DNA. Next is the G2 phase. After S phase, the cell proceeds into G2, which provides an opportunity for the cell to perform a self-assessment and make final preparations such as more cell growth, repairing of DNA, etc. as necessary before it finally heads to mitosis. Next phase is M phase or mitosis. During mitosis, there will be breakdown of the nucleus, redistribution of the DNA to opposite sides of the cell, formation of two new nuclei, cytokinesis. During cytokinesis, the cell will be split into two daughter cells. So there are various stages which are termed as prophase, metaphase, anaphase, telophase, and cytokinesis. Now we will look into the regulatory molecules of cell cycle, how cell cycle is regulated. There are several regulatory molecules which either promote progress of the cell to the next phase, which is known as positive regulation, or halt or stop the cycle, which is known as the negative regulation. These regulatory molecules may either act individually or they can influence the activity or production of other regulatory proteins. We will discuss about the positive regulation of cell cycle. There are two groups of proteins called cyclins and cyclin-dependent kinases or CDKs, which are responsible for the progress of the cell through the various cell cycle checkpoints. The cyclins are proteins that regulate progression through the cell cycle, and they are named so because their amount varies through the cell cycle. Cyclins regulate the cell cycle when they are bound to respective cyclin-dependent kinases or CDK. To be fully active, the CDK cyclin complex must be phosphorylated. After phosphorylation, they will get activated and they further phosphorylate other proteins that functions in the cell cycle. Once the cell move ahead in the cell cycle, the cyclins will get degraded. The levels of the four cyclin proteins, cyclin D, E, A and B, fluctuate throughout the cell cycle. Increases in cyclin proteins are triggered by external and internal signals, and once the cell moves to the next stage of the cell cycle, the cyclins that were active in the previous stage are degraded. There is a direct correlation between cyclin accumulation and the major cell cycle checkpoints. Although the cyclins are the main regulatory molecules that positively regulate the cell cycle, there are several other mechanisms to fine-tune the cycle. These include molecules that prevent the activation of cyclin-dependent kinases called CDK inhibitors, which directly or indirectly monitor a particular cell cycle event and block the CDKs until the specific event or error is either completed or repaired. So, coming to the negative regulation of the cell cycle, the second group of cell cycle regulatory molecules are negative regulators. They halt or stop the cell cycle. Examples of negative regulatory molecules are retinoblastoma protein RB, P53 and P21. These molecules act primarily at the G1 checkpoint. P53 is a multifunctional protein. It acts when there is damaged DNA during G1 phase. The protein P53 blocks the activity of CDKs and has been dubbed as watchman or guardian because the DNA damage in a cell is sensed by it. If damaged DNA is detected, P53 holds the cell cycle and recruits enzyme to repair the DNA. If the DNA cannot be repaired, P53 will trigger apoptosis. So, as we said, P53 is a multifunctional protein, and among its main several functions, cell cycle control is a major one. As the P53 levels rise, the production of P21 is triggered. P21 enforces the stop or halt in the cycle by binding to and inhibiting the activity of the CDK cyclin complexes. As a result, the cell will not move into the S phase. Next one is retinoblastoma protein, RB. It monitors cell size. In the active dephosphorylated state, RB binds transcription factors E2F. When RB is bound to E2F, production of proteins necessary for the G1 to S transition will be blocked. As a result, the cell will increase in size. As the cell increases in size, RB will be slowly phosphorylated and becomes inactivated, and as a result, it will release the E2F. The released E2F can now turn on the gene that produces the transition proteins, and as a result, the G1 to S transition block is removed. The cell will move forward to the S phase. So coming to the cell cycle checkpoints. What are the cell cycle checkpoints? A strong surveillance system is there in a cell to monitor the cell cycle progression itself. These are the critical regulatory points of the cell cycle, and they are termed as the cell cycle checkpoints. As the cell progresses through the various phases of cell cycle, it is through a specific and controlled manner with checkpoints. These checkpoints will ask if the cell is ready for the next step, like is cell large enough, is the DNA healthy without any damage, etc. As a result, the cell ensures that it generates healthy daughter cells. If the cell cycle runs too rapidly without the checkpoints, then there will not be enough time for the cell to enlarge, to make up enough cellular organelles, etc. And it will lead to abnormally small daughter cells. And these cells may fail to survive. Likewise, if a cell undergoes mitosis without identifying or repairing the DNA damage or mutations, then the daughter cells may turn into a cancerous cell. So, it is very much necessary that the cell cycle is governed or regulated by the critical checkpoints. So, what and when are these cell cycle checkpoints functioning in a cell cycle? The cell cycle is controlled at 3 checkpoints. The integrity of the DNA is assessed at the G1 checkpoint. Proper chromosome duplication is assessed at the G2 checkpoint. And attachment of each kinetochore of a chromosome to a spindle fiber is assessed at the M checkpoint. This image shows the various cell cycle checkpoints and the cyclins and CDKs involved at various phases. During G1 phase, cyclin D and CDK4 and 6 react to outside signals such as a growth factor or mitogen. During G1 to S phase, cyclins E and CDK2 regulate the centrosome replication. During S phase, cyclin A and CDK2, it will target the helicases and polymerases. And during the M phase, cyclins B and CDK1 regulate the G2M checkpoint. So now we will discuss about the G1 checkpoint or DNA damage checkpoint. This is the first checkpoint which regulates the transition of a cell from G1 to S phase. The G1 phase, as we said before, it is a state in which a cell reaches immediately after cytokinesis, and generally it is very long. The length of G1 phase is generally constant for a given cell type. This checkpoint is also known as the restriction point. During this point, the cell irreversibly commits to cell division. During this checkpoint, the cell will pass only if it is having an appropriate size and has favorable extracellular environment, and if it is not having any DNA damage. The extracellular environment includes nutrient availability or predatory threats and an external trigger such as a mitogenic hormone or a paracrine signal. So, if a cell does not meet these requirements, it will not progress to the S phase. The cell will be halted in the cycle and it will attempt to remedy the problematic conditions. If a cell meets the requirements for this G1 checkpoint, it will enter the S phase and begins the DNA replication. So, the G1 checkpoint checks for enough nutrients, enough cell size, is the environment okay, and if the DNA is okay. Next is the G2 checkpoint or DNA replication checkpoint. This second checkpoint regulates the entry of the cell into the mitosis. This is triggered by MPF or maturation promoting factor or M phase promoting factor. This MPF is a cyclin CDK complex. It promotes mitosis by phosphorylating a variety of other protein kinases. Here also, as with the G1 checkpoint, the cell size and the protein or energy reserves are assessed. The most important role of this checkpoint is to ensure that all of the chromosomes are accurately replicated without any mistake or damage. If the checkpoint mechanisms detect problems with DNA, this checkpoint will hold the cell in this G2 phase a little bit longer, and the cell will attempt to either complete the DNA replication or repair the damaged DNA. If no problems are detected with DNA, the cyclin-dependent kinases will signal the beginning of mitotic cell division. Most of the metabolic activity of the cell will be shut down and the cell will concentrate its resources on dividing the nuclear and cellular materials. So, the G2 checkpoint checks whether the DNA is correctly replicated, if the replication is finished, whether there are enough protein energy reserves, and if the cell is having enough size. The next is the M checkpoint or spindle checkpoint. This is the third checkpoint and it occurs during mitosis, near the end of the metaphase stage of mitosis. And this checkpoint regulates the transition from metaphase into anaphase. It ensures spindle fiber formation and that all the chromosomes are properly attached to the spindle at the metaphase plate. After attachment of kinetochores of the chromosome to the spindle fibers, the anaphase promoting complex APC will be activated. It will trigger the breakdown of cyclin and inactivation of proteins which are otherwise holding the sister chromatids together. And this will result in the splitting apart of the sister chromatids and the sister chromatids will move to opposite poles in the cell. Thus, the M checkpoint prevents cells from incorrectly sorting their chromosomes during division. So, the M checkpoint assesses whether the spindle integrity is okay, whether all chromosomes are attached to the spindle, and whether the chromosome attachment to the spindle is proper. So, what are the functions of the cell cycle checkpoints? The cell cycle checkpoints ensure that the nuclear genome is intact and is without any damage. The conditions are appropriate for the cell to divide. There are enough nutrients for the daughter cells. The genetic material is divided only once in a cell cycle and it is completely replicated. No damages or mutations are there in the daughter chromosomes, and if there are any mutations, they will be repaired by the repair enzymes. And the chromosomes are correctly aligned and oriented during metaphase and they are correctly attached to the spindle fibers. So, these are the importance of the cell cycle checkpoints. The checkpoints delay cell division until the problems are fixed and solved. The checkpoints prevent cell division if the problems cannot be solved. Apoptosis will be induced in the cell if the problems are severe and cannot be repaired. The cell cycle checkpoints accurately maintain the genome integrity of the organism. It ensures that only one round of replication happens per cell cycle. And if the checkpoints are not working properly due to any mutation, it will lead to the cancerous cell growth. The drive to proliferate and the need to maintain genome integrity are the two most powerful forces acting on biological systems. And these forces are carefully balanced through cell cycle and cell cycle checkpoints. So, we were discussing about cell cycle regulation, cell cycle checkpoints, their functions and importance. Thank you so much for listening.